Today we're doing Khan Academy's MCAT Prep Cells section cellular development questions. First question. A previously unknown cell type is discovered in the central nervous system of a laboratory strain of mice. In cell culture experiments, researchers find that the cell type can self-renew, but it can only differentiate into one other cell type. Which of the following best characterizes the cell type? Multipotent, unipotent, multipotent. Oh, I was missing the last part. Multipotent embryonic stem cell, unipotent somatic stem cell, multipotent somatic stem cell, or unipotent embryonic stem cell. You work with stem cells.
I do. And part of my paper is actually on the differentiation between multipotent, unipotent, pluripotent, and totipotent. And so the definition of all of them, I doubt you'll have to know it for the MCAT, but quickly, totipotent is the highest ability to differentiate into all of the three types of adult germ layers, so endoderm, mesoderm, ectoderm, and the placenta.
Right.
And then pluripotent would be all three of the adults germ lines, and then
But not the placenta.
right, that's the differentiation between totipotent and pluri. And then multi would just be multiple, which it is in this question, and uni is obviously one. And then the other important thing about whether or not to differentiate between somatic and stem cell to me was that they mentioned self-renewal, which is a major contributing factor to what classifies something as a stem cell.
Self-renew. So
Oh no.
is it embryonic or somatic?
So I would go with embryonic because it said self-renew. And I said multipotent.
Oh, I would think unipotent because it said one other cell type.
Wow, I got this totally wrong.
Okay, so I think the correct answer is unipotent somatic stem cell, but let's find out why. So,
because it says like differentiates into one other cell type, and then it said which of the following best characterizes this cell type. Actually, I don't know.
well, I think let's look at those hints. All stem cells must have the ability to reproduce copies of themselves as well as differentiate into other cell types.
Yes.
Some stem cells, such as the ones that replenish the skin, are unipotent, meaning that they are capable of differentiating into one cell type. So that's what they said in the question. The unknown cell type that the researchers discovered is best characterized as a unipotent somatic stem cell. So why somatic, though? It's discovered in the central nervous system.
So ectoderm.
Right.
So for me, I would have needed to hear that the other cell type other than self-renewal would have been something inside ectoderm as well.
Oh.
Did they mention that? Did I just miss it?
Well, no, but I'm getting it now. They said it's embryonic versus somatic, so you would assume with embryonic you haven't made the body yet. Therefore, you can't have somatic. But once the body is created, so they're saying like this is a laboratory strain of mice with a central nervous system, meaning it's fully developed. So it should be somatic at that point, not embryonic. Embryonic would mean that it's involved in development, but this is already past the stage of development.
Right.
So that explains the somatic thing to me. And then unipotent, they said only one other, like one cell type.
Yeah, I think I put too much emphasis on one other cell type, and they were just saying other in addition to self-renewal.
Oh, yeah.
So the biggest takeaways here recognize that self-renewal is big for stem cells. Maybe know the differences between the multipotent, the potential to differentiate into for different germ layers for stem cells, and anything else.
Well, one thing for me that I would want to point out is that I don't know anything about these.
Yeah.
You know a lot about these.
Yeah, and I got it totally wrong.
Right. That's the whole, like, fine dining and breathing. Like, leave your knowledge behind you going into the MCAT. Yeah, so that's maybe one thing that I would say. But other than that, it makes sense. You can really get a hang of that question from just the words that they use in the question. Okay, next question. Researchers compared the rate of production of daughter cells produced per differentiation event in a population of multipotent somatic stem cells that is undergoing stochastic differentiation with the rate found in a population undergoing obligate asymmetric replication. What would they find? So we're comparing somatic stem cells that are going stochastic versus obligate asymmetric replication. I don't know what these mean, but the possible answer choices are daughter cells produced per differentiation event would be the same in each group. They would be three times that of the latter, half that of the latter, or daughter cells produced per differentiation event in the former group would be twice that of the latter. So it's basically which one of these is producing more than the other, or are they producing the same as each other? Stochastic versus asymmetric. Obligate asymmetric. For some reason, that just strikes me as probably producing more. Do you have any idea on these?
I have no idea.
All right. Time for us to guess on the first.
Wow.
Okay, so I just picked a random one and I got it right. So the correct answer was daughter cells produced per differentiation event in the former group, meaning the stochastic differentiation, would be twice that of the latter. So stochastic is more. All right, so the hints are, in stochastic differentiation, a stem cell differentiates into two daughter cells. The original stem cell ceases to exist.
Right.
And a nearby stem cell undergoes mitosis to create two stem cells. So there's a net gain of two daughter cells while the number of stem cells remains constant. Wait, that... Oh, okay, I see. So let's just say you had two stem cells. One of them becomes two daughter cells. So you lost a stem cell, but then there's the one that was next to it regenerates, mitosis, creates a copy of that other one that then became the daughter cells. In obligate asymmetric replication, a stem cell differentiates into one daughter cell while also continuing to exist. There's a net gain of one daughter cell while the number of stem cells remains constant. Okay, so that makes sense with our answer. Daughter cells produced per differentiation event in the former group, meaning stochastic, would be twice that of the latter, because in stochastic you have two daughter cells, in obligate asymmetric you have one daughter cell. Next question. A team of zoologists discover that direct cell-cell communication plays a key role in the early cardiac development of monitor lizards, and that the molecules responsible for this communication are downregulated in adolescents and adults using a proprietary assay they developed in their lab. Which of the following universally known assays would most directly confirm the difference between embryonic monitor lizards and more fully grown monitor lizards? Is it ELISpot to detect secretory products of cells, northern blot to detect specific RNA sequences in a sample, immunohistochemistry to detect cell surface proteins and glycoproteins, or a southern blot to detect specific DNA sequences in a sample? So detecting a DNA sequence doesn't do anything.
Right, they would both,
should be identical.
Putatively share the same genes.
Yes, good word choice. Immunohistochemistry, cell surface proteins and glycoproteins. They're not really talking about that in here, right?
Yeah, not cells. Well, it does say cell-to-cell communication.
Maybe. And that one would make sense. So in my head, I was thinking it's got to be a transcription thing.
Yeah, I was thinking about expression too, so I clicked northern blot. But now going back and saying cell-to-cell communication, okay, and the molecules responsible for this communication are down-regulated in adolescence. It does sound like expression to me, but the molecules responsible for cell-to-cell communication might be what's on the outside, so glycoproteins and surface proteins. So
do you know anything about northern blots?
Other than that they, it's kind of an expression thing, no. Oh, and I don't think they're quantitative. Like, I don't think they could tell me, like, I would have to do a microarray to get a quantitative, or a qPCR to get a definition of, like, how much I'm making. And compare it between the embryo and the adult.
Whereas immunohistochemistry, the amount of, like, glow, or the amount of fluorescence makes a difference, right?
I'm not quite sure what it is. I've never done that or ELISpot.
I like immunohistochemistry.
Nice.
Okay, we got it right. So let's look at the hints real quick to make sure that we're doing this right. Direct cell-to-cell communication occurs as a result of antigen-antibody interactions on the surface of cells. Direct cell-to-cell communication? Okay. Antigens and antibodies on the surfaces of cells are made from proteins or glycoproteins. Immunohistochemistry assay. Okay, so they directly characterize antigen-antibody interactions. Immuno. Okay, that's the key thing there. All right, next question. Which of the following organelles plays a key role in apoptosis? Endoplasmic reticulum, mitochondrion, lysosome, Golgi apparatus.
Do you know?
No.
Okay, I was thinking lysosome because it contains dangerous things, and I was incorrect.
Yeah, I wouldn't think lysosome. That's about like endocytosis. Mitochondrion, I would assume. Mitochondrion, because Golgi apparatus and endoplasmic reticulum don't make sense to me. Mitochondrion, if your energy stops working, then you could, program cell death. Okay. That was right. All right. You see anything in the hints important?
Yeah, the mitochondria of the cell are perhaps the most essential for the immediate life of the cell. This is where the energy to power enzymes and maintain ion gradients comes from. So I was thinking maybe like something actually gives a signal, and they were just saying the signal happens because one of these stops working.
Right.
And yeah, mitochondria is essential.
Okay. All right, cool. Next question. Question number 5. Researchers hope to design an assay to measure the progression of apoptosis in live cells in real time. Which of the following 4 methods is most promising avenue to pursue in order to develop this assay? So you want to detect apoptosis in real time live cells. Detecting increases in mitochondrial concentrations of cytochrome C, measuring decreases in mitochondrial volume, measuring increases in cytoplasmic volume, detecting increases in cytoplasmic concentrations of cytochrome C. Okay, you're trying to develop an assay, so we got to think practically here. What's the most likely thing to pursue?
C sounds impossible. I don't know how I could measure internal volumes of organelles and the cytoplasm.
Yeah. I kind of like detecting increases in cytoplasmic concentrations of cytochrome C.
That is what I would go with as well.
Because that would imply that your mitochondria are like falling apart or something.
Yep.
Yeah, so I'm going to pick that.
Something that's there that shouldn't be there.
Yes. Okay, we got it very right. Oh, that's good. Something is there that shouldn't be there. Okay. All right. Next question. Caspase enzymes mediate apoptosis by attacking which of the following? So caspase. The cysteine residues in cytochrome C enzymes, the cysteine residues in cytoplasmic proteins, the aspartate residues in cytoplasmic proteins, the cysteine residues in BCL2 family proteins. Caspase. Caspase is involved in apoptosis. But how does it do it? I'm going to say cytochrome C enzymes because we keep talking about mitochondrion.
Yeah.
I don't know what BCL2 proteins are.
That's not the breast cancer thing. What's the breast cancer one? BRCA something?
Oh, yeah, I'm not sure.
Okay. So, yeah, I would go with cytochrome C as well.
Let's see. Cysteine residues in cytochrome C. It's not that. In fact, the correct answer is the aspartate residues in cytoplasmic proteins.
I kind of halfway wondered if that's what the asp part of caspase was. I don't know.
Well, either way, that's a good way of memorizing it. Okay, the hints are telling us caspase enzymes are proteolytic enzymes found in the cytoplasm. They're activated by cytochrome C. Caspase enzymes have cysteine-rich active sites that target aspartate residues in cytoplasmic proteins. Caspase enzymes mediate apoptosis by attacking aspartate residues in cytoplasmic proteins. So they get rid of cytoplasmic proteins because they have a bunch of cysteines and they can attack the negatively charged aspartate. Okay. I like your thing though, that aspartate.
Well, that's just, I think, part of the enzyme naming thing that I find so fascinating and helpful.
Okay, next question. Which of the following is not a mechanism for induction of cell differentiation? paracrine signaling, formation of gap junctions, direct contact, autocrine signaling. I think this is gap junctions.
I do as well.
Direct contact, we talked about that before, that like if two things butt up against each other, they should slow down, like in cancers, but they don't. So autocrine signaling, cell differentiation, that's hormone telling it to do something. Paracrine signaling, that is one cell telling another cell to do something. Formation of gap junctions, that sounds right. Everything else sounds wrong. Oh, that's wrong.
So that is what I put as well. I was like, oh, gap junction. And I kind of skipped over, all of the other ones. And then I did what Kevin did and assumed that autocrine signaling was something like hormones or something like that. Failing to realize that auto was, telling itself to differentiate.
Right.
So that's not something that happens.
What?
Yeah.
Well, autocrine signaling is still, it's a hormonal signal, but it's a hormonal signal to itself. Induction occurs when entities external to a cell initiate the process of differentiation. Well, that says it right there. Induction. So...
Yeah, because think about it, there would be no reason to contain in your genes okay, our whole blueprint is in our genes, but not exactly what kind of cell that cell is going to be.
Right.
It kind of gets put, it's like the soccer mom that's like, oh, or somebody that's organizing a race or something, like, we need this set of volunteers here or this set of volunteers there, to do these different functions, and they kind of get sorted out. Like, they just show up and they have all of the genes, all of the blueprint.
Right.
And then they get told where to go.
They don't tell themselves where to go because that could be a mess.
Yeah.
Okay.
Too many type A personalities in the building.
Yeah, exactly. Okay, so autocrine signaling, it needs an environmental stimulus to tell it how to differentiate. Okay, next question. Question number 8. A laboratory receives funding to investigate methods for extending the Hayflick limit in certain rapidly dividing cells in the bone marrow. Which of the following is most likely to be an area of investigation? Shrinking the length of telomeres, downregulating the production of telomerase, extending the length of telomeres, eliminating telomeres altogether. So what do they want to investigate if methods for extending the Hayflick limit? I'm going to guess that means, just based on the answer choices, that Hayflick limit is like when a telomere runs out.
Yeah.
They want to extend it in rapidly dividing cells in the bone marrow. Which of the following is best area to investigate? Well, if they're rapidly dividing, then maybe you don't want to eliminate them altogether. That's silly.
Yeah.
You want to upregulate telomerase, if anything, because telomerase would create telomeres. You don't want to shrink the length of the telomeres, so it's got to just be extending the length, I guess.
That doesn't make any... What?
Okay.
Why was this even a question?
Okay, what do the hints tell us?
The Hayflick limit refers to the number of mitotic replications a cell can undergo before it enters a senescent state, so it stops undergoing mitosis. So this is like in cell culture, we can only do so many passages before our cells just stop doing what they're supposed to.
Okay.
Each time a cell divides, its telomeres become shorter and shorter, that whole aging thing that the MCAT really seems to want us to know. Extending the length of the telomeres is one way, theoretically speaking, to extend the Hayflick limit for dividing cells. Oh, okay, yes.
Yeah, is it click now?
Yeah.
I guess I just got it through process of elimination, but yeah, it's the only one that is making them longer and not actively making them shorter.
Yeah.
All right, question number 9. Which of the following cells is not capable of entering replicative senescence? I don't really know what senescence is totally, but let's see. Which ones cannot enter it? Skin cells, cardiac cells, pluripotent stem cells, brush border cells. Replicative senescence. So you kind of understand that better than me, I think. Senescence.
Yeah, and I think this is a term that my boss uses all the time that I'm not super familiar with, but I think it's like they stop growing.
Okay.
Yeah, or maybe Yeah, that has to be it, because if they stop replicating, then it wouldn't be replicative senescence. So if they stop growing, I feel like cardiac muscle cells can do that.
They can
like to maintain the shape. Maybe they stop actively growing, but they could replicate. Skin cells.
Yeah.
Okay, so skin cells. I don't know, because fibroblasts is what I'm thinking of, but it's saying actually skin cells.
Well, for skin cells, I could see that the issue of size wouldn't be that much of an issue because we're constantly pushing out old ones.
Yeah, so I think they're a good example of replicative senescence. Like the total amount doesn't change.
But then that would mean the same thing for brush border cells.
Yeah.
Pluripotent stem cells, not... Oh, which of the following is not capable of entering replicative senescence? We might have our answer.
Is that cardiac muscle cells?
Well, no, I would say I think that your analysis of
too in depth.
No. I think you were right on that it would be like you reach a certain point and then you just maintain that point. You're not really growing or anything like that. You maintain that point.
Yeah.
Pluripotent stem cells don't really do that.
Right.
And it really seems like which of these things is not like the other. I think all of those things arise from the mesoderm. Pluripotent stem cells are kind of out in left field.
Oops.
Oh, it's not right? Wow.
Why is it cardiac?
Dang.
I guess I don't understand this. Okay. Replicative senescence is a state that mitotically active cells enter into when they have reached their Hayflick limit, when their telomeres become very short. Yes.
Okay.
Cardiac muscle cells are not mitotically active. They don't divide. Okay.
Oh.
That makes sense.
They don't divide. So unless I guess you break cardiac muscle cells or they go away, they're not dividing. You just have those cells and they just stay there.
Oh man, we gotta take care of that thing.
Dang. Okay. So a state that mitotically active cells enter when they've reached their Hayflick limit. Huh.
I might need to watch the video on that one.
Yeah, probably. But I at least understand that about cardiac cells now. Like they're not actively replicating.
Yeah.
They kind of stay still. Okay, last question. If the cytoskeletal model of cell movement is true, which of the following would one not expect to find in motile macrophages as compared to stationary macrophages, assuming all else to be equal? Increased levels of mRNA, increased levels of DNA polymerase activity, increased levels of ribosomal activity, increased levels of gene transcription. You would not expect to find in motile macrophages as compared to stationary macrophages. You would not expect... So if they're motile... I think... Okay, so we know that increased levels of mRNA and increased levels of gene transcription are the same thing.
And an increased level of DNA poly, right? Oh, no, all RNA poly would be creating RNA.
DNA poly would just be dividing.
Yeah, so I guess that would be the same.
You think... So... Wait, would
so
not
if
be...
So if I'm increasing levels of transcription, I have more mRNA, and then there's more for ribosomes to jump onto.
Oh, yeah.
Okay, so I think I might have gone with DNA poly.
Hey, we were right. Okay, yeah. So, okay, yeah, this question is just weirded strangely, but yeah, you would not expect to see any difference in DNA poly between the two because it doesn't have to do with, like, the motion.
Yep.
Of macrophages. All right, very cool. Strong work, Small.